Hello students and welcome to your next lecture in genetic innovations. In this section we will be discussing DNA sequencing. So let's take a look at the learning objectives for this section. Firstly, we will have to describe the process of Sanger dideoxy sequencing. Then compare the two methods of electrophoresis that are used in Sanger sequencing. We will then differentiate between Sanger sequencing and next generation sequencing, and then discuss the bridge amplification method that is used in next generation sequencing. Let's begin by discussing what DNA sequencing is. So we know that in our genomes we have a code, which is the genetic code, and our DNA is composed of four main bases: adenine, guanine, cytosine, and thymine. So the linear sequence, or the order in which those four bases exist in DNA, in order to determine that linear sequence, we use a technique called DNA sequencing. In the standard method of DNA sequencing, this method is unable to differentiate between modified and unmodified DNA bases, such as methylated cytosine residues. However, the technique itself can be modified or adjusted in order to understand which of these DNA bases were originally modified. DNA sequencing can also be used to sequence RNA, and like we've discussed with the case of reverse transcriptase PCR, RNA can be converted to complementary DNA using reverse transcriptase, and it can then be sequenced using the DNA sequencing technique. Now, dideoxy sequencing was the first DNA sequencing method that was described. And this method was discovered by a gentleman named Fred Sanger in 1997. For that reason, dideoxy sequencing can also be called Sanger sequencing or Sanger dideoxy sequencing. Sanger sequencing requires amplification of a DNA fragment that had first been purified using PCR or cloning. So first, a fragment of DNA can be amplified using PCR or molecular cloning, and following this amplification, it is then sequenced. In order to sequence DNA fragments with Sanger sequencing, nested sets of DNA copies are labeled and they are then separated by electrophoresis. And when we talk about nested sets of DNA, we talk about sets of DNA or copies of DNA that are made from the original PCR product or the clone. So nested refers to something within a DNA fragment. Another method for DNA sequencing is massively parallel sequencing, which is more commonly referred to as next generation sequencing. In this technique, millions of fragments are sequenced simultaneously, and this technique does not involve the use of electrophoresis. So let's discuss Sanger or dideoxy sequencing in more detail. Similar to PCR, a Sanger sequencing reaction also contains primers and a DNA polymerase. During the sequencing reaction, a PCR product or target DNA is first amplified. So multiple copies of that target DNA fragment are made. The target DNA fragment for which we wish to determine the sequence is then purified. So it's isolated or purified, and then that fragment is denatured. So when we talk about denaturation, we talk about melting the two strands of the double-stranded DNA. Following denaturation, only one primer is used to amplify that DNA. For example, if we're using the forward primer that amplifies in a 5 to 3' direction, we will then have amplification of that product from the original PCR product, since this is a nested amplification. And each of these products end up with variable lengths of 3' ends. Now, the reason that the lengths of the 3' ends vary is due to the presence of a dideoxynucleotide. So, in this reaction, we will have standard DNTPs or deoxynucleotide triphosphates, and the reaction will also contain low concentrations of dideoxynucleotides. Now, dideoxynucleotides differ from deoxynucleotides due to the fact that they lack a 3' hydroxyl group. So here we have a deoxynucleotide, a DNTP, and on the 3' carbon, there is a hydroxyl group. Now, this hydroxyl group is required for the addition of the next ribose on the sugar phosphate backbone. However, if we have a dideoxynucleotide triphosphate, there is no hydroxyl group here. And what occurs when we incorporate a dideoxynucleotide triphosphate is chain termination. So the sequencing reaction will stop once a dideoxynucleotide has been added or incorporated, and that is due to the fact that another base cannot be added when there is no hydroxyl present at the 3' end of the ribose sugar. So now that we understand how dideoxynucleotides work, let's look at how labeling and detection occur in Sanger sequencing. So each of the DDNTPs, or the dideoxynucleotides, exist at a percentage of 1% or approximately 1% of its corresponding DNTP. So for example, DDATP will be 1% of the DATP in the reaction mix. Now, the reason we incorporate such a small percentage of the dideoxynucleotides is because we would like to favor chain elongation. So we incorporate the DDNTPs at 1% of each of the corresponding DNTPs. So this will favor elongation of the chain from the 5' end. Now, what happens in this case is that DNA then have common 5' ends since we've only used one primer. So we've got one primer, in this case the forward primer. So they will have the same 5' end. Each of these fragments in this nested amplification will have the same 5' ends. However, the lengths of the 3' ends will differ depending on whether a dideoxynucleotide triphosphate was incorporated. Now, in the reaction, we'll have variable lengths of 3' ends, and these ends are then separated or resolved according to size using electrophoresis. In addition, the dideoxynucleotides are also labeled with a fluorescent dye or differently colored dye. So when a dideoxynucleotide triphosphate is added, it also emits a signal or a specific light signal depending on the base that was present at that location. So each of the 4 DDNTPs are labeled with a differently colored dye. For example, DDATP is green, DDCTP is blue, DDGTP is red, and DDTTP is yellow. So the dideoxynucleotide will emit a specific color signal depending on which nucleotide was incorporated. So the fragments from the sequencing reaction are then resolved using electrophoresis. And 2 different techniques can be used: slab electrophoresis or capillary electrophoresis, which we will discuss in the next slide. So the fragments are separated by electrophoresis, and let's say, for example, we've used capillary electrophoresis. The fluorescent signal can be detected using a detector, and as each fragment passes through the detector, a different color signal will be emitted depending on the nucleotide that was incorporated, or the dideoxynucleotide that was incorporated into that fragment. This data is then converted to an electropherogram, and this electropherogram will give you an idea of which base was present at each position. So let's look at how electrophoresis works in Sanger sequencing. So DNA fragments are separated according to size. So the resolution of the gel that is used or the capillary tube that is used for electrophoresis can differentiate a one base pair difference. So you can tell the difference in terms of read lengths in terms of a one base pair difference between two fragments of DNA. So that's a single base resolution. So the fragments are separated according to size, and here on top in part A of this diagram we have a slab gel. So as you know, agarose gel electrophoresis cannot be used to differentiate between one base pair, and so polyacrylamide gels are used in a Sanger sequencing reaction. So the samples are loaded into the wells of a polyacrylamide gel, and as you know, with all methods of gel electrophoresis, the smaller fragments will migrate faster. And so each of these little bands will have a signal or a fluorescent signal depending on the color or the nucleotide that was incorporated at that position. And since we have one base pair resolution, each fluorescent signal at each band will then indicate which base was present at that location. We can also use capillary electrophoresis. During capillary electrophoresis, we have one labeled ddNTP in each well in this reaction. So each well will contain just one labeled ddNTP. So for example, well 1 will just contain ddTTP, or well 2 will just contain ddATP, and so on and so forth. So each of these wells will just have a single labeled dideoxy NTP. And then during the capillary electrophoresis reaction, all of the samples are pooled into one reaction vial. So here we have our reservoir containing the anode, and each of these wells contain the sequencing reaction products. So the sequencing reaction is going on in these wells on a cathode plate. A capillary tube is added directly to each sample, and electrophoresis is then conducted on multiple samples at a time. So we have samples from 8 wells, for example, going into one capillary tube. Here we go, and it's going through a single detector. And that detection window also has, these capillary tubes also have a resolution of one base pair, and the laser is passed through each of them one at a time, and these fragments are then picked up on the detector. So each fragment will be separated according to size at one base pair resolution, and as each of these DNA fragments that are labeled at the end with a particular nucleotide or ddNTP will then be detected on the detector to create this electropherogram. So both capillary electrophoresis as well as slab-based gel electrophoresis can be used to separate the products during a Sanger sequencing reaction. Now let's discuss next generation sequencing. So Sanger sequencing has been a standard method of DNA sequencing for almost 30 years now. However, in Sanger sequencing, we require a predetermined stretch of DNA, and Sanger sequencing has a maximum read length of approximately 800 base pairs. Today, Sanger sequencing is used for re-sequencing small stretches of DNA. Next generation sequencing is a technique that began in 2005. And since then, the amount of DNA or data that is created per run has really increased. And when we talk about the amount of data per run, we talk about the sequencing throughput. So the throughput has increased drastically with next generation sequencing. So instead of just sequencing an 800 base pair fragment at a time, we can now sequence an entire genome or an exome in one run. Additionally, the cost of sequencing has also been reduced in this method, so it is much cheaper to use next-gen sequencing for a whole genome or large data set compared to Sanger sequencing. And as I've mentioned, next-gen sequencing can be used to sequence entire genomes or entire transcriptomes. And when we talk about the transcriptome, we talk about all transcripts that have been then converted to cDNA. And huge amounts of DNA can be sequenced simultaneously. So this graph here shows the read length against the sequence throughput in megabases of DNA. And so we have the Illumina HiSeq system here, which sequences just short fragments of DNA, 125 bases. However, it has a throughput of approximately 1 million bases of DNA. So these platforms can differ in terms of the length of reads. So here we have other systems such as the Roche system, which has a medium read length. So it sequences an 800 base pair sequence at a time, but the throughput is only approximately 1,000 bases. So as you can see in some of these new modern next-gen sequencing techniques, shorter fragments of DNA are sequenced at a time. However, a lot more DNA is sequenced. So DNA is split up into short contigs or short fragments, and each of these short fragments are rapidly sequenced, and then these sequences are aligned. Now let's look at the table which compares some of the differences between Sanger sequencing and next generation sequencing. In terms of gel electrophoresis or capillary electrophoresis are an integral part of sequencing during Sanger sequencing. So Sanger sequencing or dideoxy sequencing cannot work without gel electrophoresis. However, electrophoresis is not used in next generation sequencing. Sanger sequencing is only semi-automated, for example, in the case of capillary electrophoresis. However, next generation sequencing is a fully automated process. During Sanger sequencing, the sequence data cannot be recorded as the reaction proceeds. So the fragments can only be separated at the end of the reaction, and these fragments are separated by gel electrophoresis, and then the sequence can be determined. However, in next generation sequencing, sequence data is recorded throughout the sequencing reaction. Now let's talk about preparation of the DNA template or the DNA that's to be sequenced. For Sanger sequencing, you require a short fragment of DNA that has been amplified by PCR or that has been cloned into a vector. However, for next generation sequencing, DNA fragments are ligated, the T is missing there, to an adapter, and this adapter contains oligonucleotides, which are then amplified by PCR. So a lot of different DNA fragments are ligated to these adapters, so we don't need amplification of a specific region of the genome. Multiple DNA fragments or a whole lot of DNA that has been extracted are each ligated to an oligonucleotide, and each of these fragments are amplified by PCR using a primer that can bind to the oligonucleotide. During Sanger sequencing, we can have a maximum read length of 800 nucleotides. However, during next-gen sequencing, you can look at between 35 and 14,000 nucleotides in one read. In Sanger sequencing, just one DNA template is looked at or sequenced at a time. However, in next generation sequencing, we have tens of thousands to billions of DNA fragments that are all sequenced simultaneously. The throughput for Sanger sequencing is approximately 96 sequencing reactions per run. So if we remember that capillary electrophoresis reaction and we looked at that plate, you can just have a maximum of approximately 96 reactions that occur per run. So in one run, an approximate amount of 80 megabases of DNA can be sequenced. However, during next-gen sequencing, approximately 80 megabases to 1,000 gigabases can be sequenced per run, and that depends on the sequencing platform that is used. The error rates are very low in Sanger sequencing, however, they are much higher in next-gen sequencing. So because we have higher read lengths, we do have a higher error rate for each individual fragment. However, multiple reads are conducted at the same time, and so this can give an idea of the consensus sequence by reading multiple different sequences and then overlapping these regions to determine or extrapolate the sequence data.